Welcome to Deadpool's dysfunctional biology class. You'll hate it here. You wanna know what enzymes are? I forgot what they are. You wanna know what enzymes are? They're proteins with specific shapes held together by strong amino acid bonds. Also known as squiggly meat noodles. Don't write that in the exam. Do not write that in the exam.
They're biological catalysts that speed up reactions remaining unchanged themselves. Or in less fancy words, they're your relative from 15 years ago who helped change your diaper quickly and never came back. Except the enzymes actually come. The enzymes speed up all metabolic reactions, or else you'd still be trying to digest that biryani from 5 years ago. Now, the active site is a part of the enzyme that determines what substrate can and will fit into it to be broken down. Only the right shaped loser gets in. Everything else denied harder than our will to live during the CAIs. A substrate will always be complementary to only one specific enzyme. I made this very high quality animation so you could understand better. I hope it helps a lot. In a solution, the enzymes and substrates will randomly collide at the active site. The animation is to help you know how that looks like. Anyways, the substrate is then broken down into products. For example, starch into maltose. And then the enzyme will keep going and breaking down more substrates, just like a protein hitman.
Enzymes are locks because they each have their own unique shape of pins or active site, you could say. And the substrate is the key because they each fit into their own active site or own set of pins or own enzyme, whatever you want to say. Look, boom, bam. Would you look at that product form? Pow. Enzymes work most efficiently at optimum temperatures, 37 degrees in the human body. Hot.
As the temperature increases, the enzymes and substrates gain more kinetic energy and collide more to make more products.
Or, in my words, more heat equal more movement equal more boom boom.
If the temperature gets too high, the enzymes' bonds break down and deform the active site, also known as the enzyme denaturing. It's completely irreversible. So, congratulations, you now have a useless lump of protein. Like me on a Monday.
Enzymes also require a specific pH to function, just like temperature. For most enzymes, it's 7. And for some, which are formed in acidic bases, just like your stomach, they need an acid trip, and the acidic ones need 2. Now, at a low temperature and a low pH, the enzyme won't be denatured. It will be what you call inactive, aka it's going to be lazy as shit. That's also basically me just describing myself, but now amylase is an enzyme that's found both in your mouth saliva and in your small intestine. It breaks down starch into glucose. Then we have maltase, which is also found, all of these are found in your small intestine. Maltase breaks down maltose into glucose, sucrase breaks down sucrose into glucose and fructose, lactase breaks down lactose into glucose and galactose.
Now the pepsinogen, not to be confused with Pepsi, is generator for pepsin in the stomach. The HCl in the stomach provides the optimum pH for the pepsinogen to activate and produce pepsin. The pepsin turns proteins into amino acids and peptides. Now in the pancreas, there's lipase, which breaks down fats into fatty acids and glycerol. I think I need to overdose on lipase by now. There's also these, which I don't know if they're part of our syllabus, so I kind of just left them out, but I'm putting up the picture of it anyways in case you want to learn this. Yeah, I'm a great teacher, trust me. Lastly, these are the experiments. This is the one to find out the effect of temperature on the enzyme. I got a little lazy at this point, so I've just put it up as the picture so you can read it. You can pause and you can read the effect of how to investigate the effect of pH on the enzyme. Please rate me 5 stars. Please.